So in the next session or next part of the session here, I'll be talking about finding, accessing, and citing data. So for starters, if we can just step back and reflect on our experiences of searching for something on the web, finding data online is a bit different than searching for documents and web pages. But when it comes to text documents and web pages, a typical search engine like, say, Google does a great job of finding something relevant to your free-form keyword search. However, if you're trying to locate something very precise, especially if it's not a document or web page, generally it doesn't quite work as well. In some ways, searching for data is a lot more like online shopping, where you're looking for something specific, and often it's a physical object like a car or clothes. Again, just reflecting on our own experiences searching the web. Let's take, for example, the search text here for a 2018 or newer Mazda, something very specific with less than 20,000 miles, and et cetera. And, just imagine how well that, how well you think that would work for you. Um, so the alternative are sites specifically dedicated to shopping or specific to a type of product, like a car or something like that. And when you're doing online shopping, you're actually encountering a technology that's called faceted navigation. And in simple terms, this is your typical search and refine experience, where you might start by entering a keyword search and then refine with the filters by category to locate exactly what you're looking for. So FaceBase follows this type of faceted navigation or search and refine paradigm for finding data. And we'll see in a moment as we move to the online demonstration that the FaceBase homepage has a search bar at top uh, or a browse all data link. And then when it presents the results, it gives you a number of filters by various categories relevant to the data. Now, it's also important to pause for a moment and also know that face-based data are organized in a structured and intuitive data model. So this helps to ensure that face-based data are ready for reuse, for reproducibility, and for analysis or reanalysis, as the case may be. So just to introduce you to this model, the structure of data and FaceBase, going from top to bottom, a project is a general term that we use for any kind of group that is contributing data to FaceBase. So a project will have a funding acknowledgement, usually an abstract on what their overall research agenda is, a list of team members and PIs, as the case may be. So we don't use a more specific term because it could be, say, intramural research group. It could be an externally funded, like an RO1 single investigator, or maybe a collaboration. Could even be a consortium of groups, or just an individual lab. So we don't try to define it further than that. We just call it kind of a project. But these are the groups that are contributing data, and then beneath that are data sets. So one project may be conducting an overall study, and they may produce more than one data set. So a data set could be one phase of an overall study or funded research activity. You might have a project that releases a series of data sets that are kind of interrelated. Maybe they cover different cohorts or different syndromes or different areas of focus. And then within a data set, we have descriptive metadata or information about the data called metadata on, say, the experiment. This would cover, or sorry, it could be multiple experiments. So these are kind of like finer grained. So each assay type that was conducted, including details on the protocols used. We generally take a protocol document or maybe a reference to a published protocol, so you don't have to repeat all of it in our database, but that you can provide the information so that other consumers of the data, as you're looking for data, you'll be able to find the protocols that were relevant to the data within each data set, and you can drill down into data acquisition or specimen preparation and find out more of it. Our more recent data has better coverage of the protocols because this is something that we kind of focused on starting several years ago, and FaceBase was established in 2009, so we go back pretty far. But even the older data generally has references to publications that will describe protocol details. Okay, and then for each experiment, there's generally going to be a number of replicates if it's an animal model-based study. So you'll have biosample characteristics of, say, mouse or zebrafish or other model organism, that, as the case may be. We will also have sample details for human subjects research or demographics information, and these are also located at the same level in the hierarchy, and then the actual data files. So FaceBase has both open access as well as controlled access data. Our open access data is available directly from our website. Our controlled access data requires that you go through you submit a data access request that is reviewed by an NIDCR data access committee, and Alejandro next is going to show the process for that. But most of our data that's our data that's open access, it's all available just directly from the website. You can just click and download. So this structured information is what powers the search and browse experience behind the scenes. Okay, so when you do find data of interest, then you drill down to a data set individual record or the details on that data set. And you can view its detailed descriptive information, aka its metadata that describe the data. On the right-hand side is a large thumbnail of just the top part of a particular data set detail page. In the highlighted region, you see the most high-level descriptive information, like its title, description, and attribution details. In particular, I'd like to point out that every data set is given a digital object identifier, DOI, just like any journal publication. So this ensures the long-term citability of FaceBase data. And when you use FaceBase data, you include these DOIs in your citations, and I'll talk about data citation later. But this way, someone will know exactly what data you were referring to that you used in your in your own analyses. So data sets also include, as I mentioned, protocol documents for the in-depth explanation of the experimental procedures that produce the data. Then a set of keywords such as experiment type, or that's the different types of assays, for example, or imaging modalities used, the species, because we do have multiple species, as well as human subjects, and then anatomical regions, phenotypes, syndromes, and several others to help categorize the data. Again, this is to ensure the findability of data. And these typically come from a standardized terminology. So if a term exists, it'll come from a standardized terminology. We look for those terms that have been contributed to public ontology sources, as they're called. And this way, the data are described consistently, even across different data sets produced by different contributing projects. Now, you can, as I mentioned, you can download all of the open access FaceBase data right from any browser, and you don't even need to log in. So there's no special software required, and there's not even login required. So it's a really simple process. You find the data, you want to check it out, you download it. There's no software to install, no tools to run. However, if you download, say, large collections of data, say for a large sequencing experiment or even a large high-resolution imaging data sets, it could be tedious to download them one by one using your web browser. And of course, high-resolution images could themselves be a gigabyte or more, and certainly raw sequencing reads can be a gigabyte, can be 10 gigabytes, even larger. So it's not really something that you probably always want to have to do through your browser. So to make it easier, we do have a desktop and command line tools that you can install. And then what you do is from your browser, you download a small manifest file. Basically, it has kind of the structured descriptive information, it has all the metadata, and it has a set of links to all of the files. And then you use our download tool, um, and you do a step called materialize the data set. So what this does is takes those links and then it fetches all of the data, and it does it in a robust and efficient process. It can do retries, restart on failed downloads, and it also does integrity checks as it goes. So, when you're talking a gigabyte or 10 gigabytes, you want to know for sure that the data that you downloaded that's on your disk exactly matches what the contributor submitted to our site. So we have tools for ensuring that. Okay, and then you can also access any and all data from the FaceBase site using our programmatic APIs in Python and R programming languages. So there are additional means if you're doing other kind of analyses and you want to be able to programmatically fetch data or query really any of the data or metadata on our site, you can do that from our APIs as well. And then once you've used or reused FaceBase data in your own research, of course, you should cite the data sets that you used. And this is just as important as citing publications as a way of recognizing the research that you're building on. There are guidelines offered by major publishers. So we recommend following Nature or Science or your particular publication venue if they have their own guidelines. For every data set in FaceBase, you'll see a share and cite button in the upper right-hand corner, and click that and you'll see the share and cite dialog exactly like seen here in the screenshot. For citation purposes in your own works, we provide both a formatted data citation following Nature's guidelines and a downloadable BibTeX file. If you're familiar with that, it's a standard that you can, or it's a kind of a de facto standard that you can directly import into practically any reference manager software. So every data set has these. They include all the proper attributions, the digital object identifier. You can use these in your citation section. You can include them in the any statement about your data usage in publications. And also we have online guides on our website that provide more information and links to publisher recommendations to help you with that. Okay, and with that, I'm going to switch gears here over to a live demonstration. And let me just find the right browser. And I've kind of queued up a few things, but I'm going to start with the general search. So Chris walked you through some of the key parts of our website. I'm going to focus here on searching for data. So first of all, we have a data top level menu on the FaceBase toolbar. And if I open that up, you'll see it has a variety of categories. So there's data sets. That's your primary interface when looking for data on FaceBase typically. But we also have projects. You might be interested in a particular project that produced data that has shared their data through FaceBase. You can look directly for projects there. We have a listing of all protocols that are recorded in the system. And then we have a direct imaging search. So imaging is just one of the types of data. Of course, we have imaging, we have a large collection of sequencing data. We really have a broad array of different kind of experiment types. So we wouldn't want you to think of FaceBase as only imaging, but we have imaging, sequencing, and other related types of data, as well as in our human subject repository. We also have some more recent data that includes public health information, questionnaires, survey kind of data. So our collection of other types of data are also expanding as well. All right, so, and then you can kind of drop down directly if you want to search just within mouse, human, zebrafish, et cetera, or by specific types of experiment or modalities. You can do that directly. All of these go really into the same location. So there's not like data hiding in different corners. You find it all from the same unified search, but we think it's helpful just to have some links to drop you down directly. So there's a keyword search at the top, and then there's browse all data sets here. Now, you can just drop straight into the filter experience if you don't have any search keywords you want to begin with. But let's just say we know we want to start with my favorite keyword here, mandible. Okay, so we drop into our search experience, and now we see the mandible term repeated up here in the top search box. And then on the left-hand side, we have the refine search options, and these are all of the categories of filters. There's a couple things to know about it. One, there's nothing special about the ordering here, and there's nothing special about some things being open or closed. We just don't really want to overwhelm you on the first time coming to the site, having everything on the left side open. As I showed that information model, that overall network of the different boxes with the project, data set, and experiments, those are all kind of being mined here on the left-hand side. And that's why we have really a rich assortment, excuse me, of types of categories that you can filter on. And then, excuse me, and then, so we can search on any of these in any order. You don't have to go in the order listed here. The other thing to note is that, okay, so you can expand them. So let's say we wanted to look more at gene information. So I expand gene, I get a set of gene names here. And again, the data is really, really expansive. So we don't necessarily want to show or couldn't show every gene listed out here. So you'll see these show more buttons, links on the side. So if I click on show more, now I can search through all the genes that are have been reported in FaceBase, and it looks like we have there's 35 in total. I can expand the displaying the first, so I'm showing 25 of 35. Sometimes this list can be very long, so we don't want to show too many, but I can expand it out to 50 and just kind of browse through everything right here. I could search and filter this directly. So let's just say I'm looking for a particular, let's say I'm looking for the BMP2 gene. I find that in my list, check it, submit that, and that becomes a new filter for my data set, for my data set search. And then if I want to back out of that, I can either uncheck it directly in the box, or I can find at the top, there's a set of filters. This will grow. Right now, it just has the gene, but it'll grow as I add to it. It has a little X. I can clear that. And now I can get back to searching everything within mandible. And again, there's a little X here. I could clear that out as well. But let's just go ahead and take a look for, let's say I want to zoom in on microscopy. And actually, let's do this. Sorry. Let's just look directly here. So I typed, searched in this little category for experiment type, and go ahead and check on fluorescent microscopy. I can see a few options here. Let's say let's say instead I was looking for some kind of sequencing. So I'll clear that and look at, let's look for some type of seq data. Okay. Now, again, the show more details to see what else would appear here. Let's say an RNA-seq assay. I check that, submit that. Now I have RNA-seq assays displayed, and I can drill down into those as well. So let me go ahead and open up one, and we'll get to a data set display. So I picked this first data set. And again, what you see at the top is the title, description. This has sort of an overview of the study design, and it still has a detailed protocol listed here. I would follow this link if I wanted to see the specific details of the protocol. I'm not going to follow it right now, but that's what you would do. Each of these are blue underlined when you float over it links. I can see the contributors to it. And then the high-level keywords, RNA-seq assay, mus musculus, it's GAS1 gene, it's embryonic day 14, and so forth. And then as I scroll down, I see that it has 2 experiments that were recorded. And I can even see a detail of what was kind of a summary of what was going on here. They're both, of course, mice, same stage, and there's different anatomical sources that are being reported here in this. So we have an experiment on two different types of tissue, two different types of specimens. And then we have a list of the biosample details. We can drill down into these as well. I see that they're all wild type. And then the sequencing data. And then if I want to export this data, there's an export button over here in the upper right-hand side. It has a link. I can either get it as, I can get this as a CSV. So that's more useful if I wanted to just wanted to record some metadata, maybe to come back to later, or especially on, say, search results. But on a data set, what I really want here is to get the BD bag format. And this is the export format that you download, a high-level manifest file. It's in this BD bag format, and then you materialize it with a tool. Or if I wanted to, I could just click on these FASTQ, gzip links, and I could download a gigabyte or so at a shot through my browser if I wanted, and it does work. But it's probably not how I'd want to download 10 sequencing files if I had my choice. And then there's share and cite. And so let's open up the share and cite. I can see just how to cite this, or I have a link here to download the BibTeX file. Okay, so let me step back, find my way back, and let's just take another look here at microscopy. Okay, let's look at microscopy. Let's go ahead to fluorescence microscopy and look at an imaging data set. And here is, we'll pick the first one off the list. And okay, just finding it in my tabs there. Okay, so now we have another data set. We see again the title of it, the digital object identifier reference here. So even if you want to just say, I want to pass this along to a colleague, say, check out this data set without having downloaded it, we would recommend using the DOI. Just copy that, share it with someone, and that's a reliable way of always getting back to this data set. And of course, citing it, that's critical. And then again, this has a high-level explanation of the study design, as well as detailed protocol documents. We see the contributors to it and so forth. And this one has a publication in PubMed, so we could actually go off and learn more about it from its publication as well. And then the keywords explaining the overall structure of it. Now, it's nice to go to an imaging experiment too, because our data set, because we get to see some of the visuals of a data set. So we have a thumbnail in this case, and then details on the experiments again, followed by the biosample details, and then we have a list of the imaging data. And with that, we have the thumbnails appear here, which is kind of nice to give us an idea of what's in this data. Now, it's not just an important thing to know is that it's not just that you have this list of here's the images, go figure out what biosample it relates to. There's a structure here. Everything is linked. So if we go back up to the experiment and let's drill down specifically into, let's say, this first micro CT data. So I'm going to click on view details. And now I see that this particular experiment, oh, and here it has, as it should, a protocols document associated with it, tells us it's micro CT, and we can see that there was one biological replicate in this. It looks like it was one of the controls given the genotype. And then we see the imaging data for it. So we can see very specifically where the data came from. And in fact, if we had multiple biological replicates under this experiment, we could drill down into them, as we can here, and see exactly what files came with which replicate. So of course, if you were going to take raw sequencing data and run it through a pipeline again, you would need to know exactly what is the grouping, which reads go together, for example. And you don't want to have to do that based on a fancy file name. You want to know from very explicit metadata. All right, so let's now just drill down into one of these images, and we have an automated imaging pipeline that will process a large amount of our imaging data, and most of it is available in this manner. Okay, this one's loading a bit here. All right, here we go. Um, and so this is I'll show you a higher resolution. This is just anything that comes into our system, we process it. You can't load a TIFF in a web browser, and sometimes a TIFF image can be large. This one's not particularly huge, so I'll have other examples of that. But that's just as an example, you can drill down into any of the data in FaceBase. Okay,
And
so
Rob, just to let you know, about 1 minute left.
okay, thank you. All right, so let me just kind of give you an overview, and then I'll turn it over to Alejandro. An overview of some of the other visualization abilities. So under data, I mentioned that there's also imaging search. You can drill down directly into imaging search. So I have that page open over here to kind of cheat. And we can still filter everything based on the same terms. Again, you're just kind of mining this information space that exists within FaceBase. And so we can search through anatomical terms, stages to find just images of interest. And then when we open those up, we can see the image online, or we can download it. So you usually probably want to download to your machine, but things that go through our automated pipeline, we can actually browse throughout the Z dimension, and if it's multi-channel data, you can even do some of the adjustment and of it right online. So you can actually do this interactively. I'm just kind of playing around with this randomly to show you, but you can make adjustments that are appropriate for what you're trying to look for. And this all just kind of happens online. So that's our image viewer. Other types of visualization that are available, and these are kind of on a case-by-case basis within particular data sets. For example, some of our data sets also have online 3D volume data. So if you can't download it, sorry, if you can't, you can always download it. But if you want to just be able to see a preview of it online, we have both orthogonal slice viewers as well. The viewer has a mode that you can switch into 3D. So you can view it in both ways. I can switch between them. And then for imaging data, Alejandro later will show how you can add annotations. And so if someone has added annotations, you can also view those online. I mentioned the image viewer with multi-channel support. Let's see what we have in here. Okay, surface meshes when those have been uploaded as well. So if we have segmented data, we can show that online. And then we have an integrated genome browser. So sequencing experiments that have gene annotation tracks added, those are visible both online on the dataset as well as if you go directly through the UCSC site. And then let's see, lastly, another pipeline we have is for producing single cell visualization. So when we have single cell data uploaded, we can process it and produce these online interactive displays using the UCSC UCSC cell browser. So, and these are all, this is a fully functional UCSC cell browser over this data. So it's interactive, and you can switch modes and so forth with it. So with that, all right, thank you. And I will turn it over now to Alejandro.